Hello everyone, my name is Oceanna. My project is titled Quantifying Orexin Inputs to the Intergeniculate Leaflet, and it was supervised by Dr. Michael Antle. For some background, almost all living organisms have daily patterns in physiology and behavior called circadian rhythms. These include your sleep and wake cycles. They're generated by your body's master clock, which is a structure in the brain called the suprachiasmatic nucleus, or the SCN. The SCN controls your rhythms and synchronizes them to environmental cues. The most important cue that controls your rhythms is light, but the circadian system is also sensitive to non-photic cues such as food and exercise. Compared to photic cues, the brain structures that carry non-photic information to the SCN are much less understood. One structure in the brain that appears critical is the intergeniculate leaflet, the IGL, and the thalamus. However, we're not entirely sure how the IGL is activated and how it communicates to the SCN. It's suspected that it uses orexin to communicate, also known as hypocretin, which is an arousal neurotransmitter that is produced in the lateral hypothalamus, or the LH. Orexin also seems to play a role in preventing narcolepsy, which is a sleep disorder characterized by inappropriate bouts of daytime sleepiness. The specific characteristics of these orexin cells in the LH that project to the IGL is unknown. That brings me to my research question. What proportion of total orexin cells in the LH innervate the IGL? I hypothesized that in Syrian hamsters, retrograde tract tracing will tell me that only a proportion of orexin cells project to the IGL, but not all of them. Answering this question would help characterize non-photic circadian neural circuitry, as well as further assist in our understanding of narcolepsy as it relates to orexin. To test my hypothesis, I injected microspheres with distinct green fluorescent tags into the IGL of hamsters. These microspheres enter the ends of orexin cells and are carried back to the cell body, permitting us to track orexin cell projections to their sources of input. I collected brain slices containing the IGL and LH, and I stained them red for orexin, and then I performed an immunohistochemical analysis. First, I wanted to make sure I actually hit the IGL. Then I counted orexin cells that were microsphere negative and microsphere positive. This is the results of my immunohistochemical analysis. This image on the left is an example of an IGL. This IGL is right here, and this cluster of green are the microspheres that were injected into the IGL cells. The same cells are being innervated by these red spindly fibers, which are the ends of the orexin cells from the LH. The image on the right is the LH. It's an example of what I used to count. An example of a red cell, orexin cell, without beads, meaning it does not project to the IGL, would be this one. An example of an orexin cell that does project to the IGL, meaning it has microspheres, would be this one. This was the results of my quantification. I found that the proportion of orexin-positive cells in the LH that project to the IGL, meaning they had microspheres, was about 44%, and about 56% of orexin cells in the LH did not have microspheres, meaning they did not project to the IGL. To conclude, I found that a subpopulation of orexin cells innervates the IGL. This aligns with the fact that orexin is one of the many sources of arousal input to the IGL, and the IGL must receive inputs from other parts of the brain. This also aligns with the fact that orexin has other roles in the brain other than circadian responses, such as regulating appetite. The presence of cells in the LH that were orexin negative and microsphere positive also point to the existence of other LH cell types that project to the IGL. Future directions would be to look at behavior of these IGL projecting orexin cells to see how many are activated by non-photic treatments. Next, we know that the SCN is also innervated by orexin cells in the LH, so it would be interesting to see if it's the same or different populations of orexin cells that project to both SCN and IGL. Lastly, it'd be super interesting to see if these IGL projecting orexin cells are the ones that are found destroyed in narcolepsy. These are my references. Thank you so much for listening. I would also like to acknowledge Dr. Michael Antle and my lab members for teaching me and helping me make this project happen. I'd also like to thank Alberta Innovates for funding this project with a summer research studentship.